Hello everyone, this is the sixth lecture on carbohydrates, and today we will talk about the heteropolysaccharides. Heteropolysaccharides are made up of two or more different sugar residue. Like we have already discussed about homopolysaccharide. In homopolysaccharide, only single type of sugar are present. For example, we discussed about cellulose, glycogen, all these are polymers of glucose only. So they were homopolysaccharide. In heteropolysaccharide, we will talk about the polysaccharide, which are made up of two or more different sugar units. They are classified based on their composition, function, and occurrence. So in different organisms, different type of heteropolysaccharides are present. For example, peptidoglycans, which are present in bacterial cell wall, agar, which is present in some species of red alga like Gelidium and Gracilaria, Glycosaminoglycans, which are present in vertebrates, and hemicellulose, pectin, mucilase, these are present in plants. In this lecture, we will talk about glycosaminoglycans. In short, we call them GAGs, or they are also called as mucopolysaccharides, MPS. So what are glycosaminoglycans? These are made up of combination of three words: glycose, aminoglycans. Glycose means sugar, amino means amine group, sugar containing amine group, and glycans means polysaccharides. So they are made up of repeating disaccharide units. So what are these repeating disaccharide units? First sugar will be uronic acid. Not always, but in most of the cases, uronic acid will be there, and second will be amino sugar. So let's first talk about what is uronic acid and what is amino sugar. So when glucose and sixth carbon of this glucose is oxidized to carboxylic acid, they form D-glucuronic acid. Here, since this hydrogen at physiological pH, this donates its hydrogen, so we always use the term D-glucuronate because this is carboxylate ion present here. At this position, if this is present up the plane, then it is D-glucuronate, or we can say D-glucuronic acid. And if this carboxylate ion is present down the plane, this is L-iduronate. So there are two different types of uronic acid. First one, if this is carboxylate ion is up the plane, this is D-glucuronate, and if this is down the plane, this is L-iduronate. What are amino sugar? So if the glucose or any sugar have amine group present at this second position, the sugar has this amine group at second position, they are called amino sugar. They also contain acetylated amine group, like at this second position this amine group is acetylated, so they have acetylated group. Some contain sulfate, like here this sulfate group is present, and carboxylic group, which we talked in glucuronate and iduronate. And this sulfate and carboxylic group provide the negative charge, and negative charge attack water, and because of that they form gel like a structure that is viscous solution or mucus solution. That's why they are called as mucopolysaccharides, and they interact with proteins to form proteoglycans. Not all glycosaminoglycans interact with proteins, but most of them interact covalently with proteins and form proteoglycans. So what are the function of glycosaminoglycans? Most of the glycosaminoglycans act as ground substance of extracellular matrix. So for that in detail we will discuss in later section what is ground substance and how they work. So let's talk about the example of glycosaminoglycans. First one is hyaluronate or hyaluronic acid. This is, since here this carboxylate ion is present, we call it hyaluronate. If carboxylic acid is present here, we can call them hyaluronic acid. Hylos means glass. They are named hyaluronic because of their appearance like glass, and transparent, translucent appearance actually. So here in their disaccharide unit, they have uronic D-glucuronic acid, that is D-glucuronate is present as uronic acid because this carboxylate ion present up the plane. And second sugar, that is amine sugar, present here is N-acetyl D-glucosamine, because here this amine is acetylated, this is N-acetyl D-glucosamine. And bond between this disaccharide is, since here this OH is up the plane, this is beta sugar. So first carbon is involved in bonding with third carbon of this N-acetyl D-glucosamine. So this is beta 1,3 linkage. But in repeating sugar, next sugar unit, this first carbon of this N-acetyl D-glucosamine will interact, this first carbon will interact with this fourth number carbon of D-glucuronate. So they have different bonding between D-glucuronate and N-acetyl D-glucosamine. Beta 1,3 linkage is there, and N-acetyl D-glucosamine, when it will interact with next D-glucuronate unit, the bonding will be different, and here the bonding will be beta 1,4. And hyaluronic acid is made up of like this, approx 50,000 repeating disaccharide units are present in hyaluronic acid. So it is really a very big molecule. So what are the function where it is present? Hyaluronic acid is present in synovial fluid of joint, and it provides viscosity and lubricates the joints. What happens in our joint, if you move the joint and this space is not present and this gel is not present here, this both the bone will friction with each other and it will cause pain. So this fluid actually provided here for as a shock absorbent, and it's resist to the compressive forces. Next one is vitreous humor of the eye. So this is the vitreous humor which is present between lens and retina, and this vitreous humor give the eye is shape of the eye, and also it act as the shock absorbent to the mechanical shocks. It is present around the ovum. So this is the hyaluronic acid layer present, this is the ova, and this surrounding layer is made up of hyaluronic acid. So it actually protect the egg. Hyaluronic acid does not form proteoglycans, means it does not attach with proteins covalently. It exists as free polysaccharide in extracellular matrix. Hyaluronidase is an enzyme which actually degrades the hyaluronic acid. So it is present in semen, snake venom, and bacteria. In semen, when a sperm want to fuse with ova, it first will degrade this hyaluronic acid layer, this transparent layer. So for that, semen need hyaluronidase enzyme which degrade this layer, and then semen can fuse the ova. Similarly, snake venom have many snake venom have this hyaluronidase enzyme which degrade the hyaluronic acid in our skin, extracellular matrix of our skin, so that venom can, other component of venom can easily enter into the skin. Similarly, bacteria, to increase their infectivity, they also have some species of bacteria have hyaluronidase enzymes present. Next function is they are present in extracellular matrix of skin and connective tissues. Connective tissues like bone, cartilage, tendons, ligaments, and adipose tissue. So this is the figure of extracellular matrix. These are the different cells. So our extracellular matrix contain the collagen. This is collagen fiber. This thick rope-like structure, and this is elastin fiber. These fibers provide the flexibility and tensile strength to the tissues. So this collagen are not hanging around, actually spaces between this cell and this collagen and elastin fiber is filled by hyaluronic acid because they form gel like a structure. So they are, since they are holding these cells and collagens, elastin fiber, they are called ground substance. They are actually holding these fibers. So this is the long rope like structure. This is hyaluronic acid. These exist as glycosaminoglycans. But if you look here, this hyaluronic acid is attached not covalently, but it is linked with other proteins and these heteropolysaccharide parts. So these are proteoglycans, and in these proteoglycans other glycosaminoglycans are present as proteoglycans, and these are chondroitin sulfate, dermatan sulfate, and keratin sulfate, which we will talk about in next slides. So next one is chondroitin 4 sulfate. It is present in cartilage. Word chondros means cartilage. It provides strength to cartilage, tendons, ligaments, and to the wall of aorta. So here, as uronic acid, D-glucuronate is present, and second amine sugar here present here is N-acetyl D-galactosamine. In hyaluronic acid, we have D-glucosamine. Here we have galactosamine 4 sulfate, because this fourth number carbon has here this here it is attached with this sulfate group. So here the bonding between this first number carbon, this is beta sugar because OH is up the plane, is forming bond with this third number carbon. So this linkage is beta 1,3 linkage. When this N-acetyl D-galactosamine 4 sulfate will bond with next sugar, that is D-glucuronate, it will form beta 1,4 linkage, because this first number anomeric carbon of this N-acetyl galactosamine 4 sulfate is forming bond with this fourth number carbon of D-glucuronate. Next one is dermatan sulfate. Word derma means skin. It is found in extracellular matrix of skin, blood vessels, heart valves, tendons, and lungs, and it provides flexibility and tensile strength to tissues. So if you can see here, this is the right atrium and right ventricle, and these valves separate these two. Here similarly, left atrium and left ventricle, here we have bicuspid valve, and here we have tricuspid valve. So these valves have dermatan sulfate. Here this they also exist as proteoglycans. So this central region is protein, and this hair like a structure are actually the heteropolysaccharide dermatan 4 sulfate. So they provide flexibility and help in movement and provide the tensile strength to the tissues. Next one is keratin sulfate. Kera's word means horn. This is present in cornea, cartilage, bone, and horny structures such as dead cells, horn, hair, hooves, nails, and claws. So here the instead of uronic acid, we have D-galactose is present here. There is no uronic acid present in keratin sulfate, like we discussed there in chondroitin sulfate, dermatan sulfate, hyaluronic acid. Here we have D-galactose. And second sugar is N-acetyl glucosamine 6 sulfate. Here this amine is acetylated, and here at 6th position we have sulfate. So this bond is between galactose, first carbon of galactose is forming bond with this fourth carbon, and here in with next sugar, that is N-acetyl glucosamine 6 sulfate is forming bond with D-galactose in beta 1,3, first carbon here involved, and in bonding with this third carbon of galactose. So here the bond is beta 1,4, and here the bond is beta 1,4. beta 1 3. So when the galactose is joining with N-acetyl glucosamine 6 sulfate, it's linking with beta 1 4 linkage, and when this N-acetyl glucosamine 6 sulfate is forming linkage with next D galactose, it forming the beta 1 3 linkage. Next one is heparin. So we discussed that glycosaminoglycans are part of extracellular matrix and they provide flexibility, and they act as shock absorbent. But here heparin is different. It is natural anticoagulant and it is produced by immune cells, that is mast cell, and released into blood and is bind to the protein antithrombin. Because thrombin is a blood clotting protein, so it inhibit the thrombin, so it stops the coagulation of blood. So heparin is made up of D. First is it is uronic acid here present here, which is D-glucuronate 2 sulfate, because at second position this is sulfate is present, and here this is up the plane, so this is D-glucuronate, and it is forming bond with N-sulfoglucosamine 6 sulfate. N-sulfo, this amine is attached with sulfate group, and here at here also at third position we have sulfate group, here at sixth position. So many sulfate groups are present. So heparin is very high negative heteropolysaccharide, and bonding is similar here. When D-glucuronate is linking with N-sulfoglucosamine 6 sulfate, it's forming alpha 1 4 linkage, and when this N-sulfoglucosamine is forming bond with next D-glucuronate 2 sulfate, it's also forming the same linkage, that is alpha 1 4. So this was all about the glycosaminoglycans. Thanks for watching. See you in the next video.